Assalamualaikum, this is Nan and today we're going to talk about the gluconeogenesis. We're going to talk about the steps of the gluconeogenesis, the steps which are different from glycolysis in gluconeogenesis, and we are going to discuss the regulation of the gluconeogenesis along with those exceptional steps. And lastly, we're going to talk about the summary of the gluconeogenesis. So let's start. Gluconeogenesis. Gluconeogenesis is the reverse of glycolysis in which we move from pyruvate to glucose. It occurs in liver primarily and epithelial cells of kidney along with intestine. Now the role of gluconeogenesis is to maintain the steady glucose level. Now steps of gluconeogenesis. The steps of gluconeogenesis are almost same as glycolysis but in the reverse direction. Let us see the steps. These are the steps of glycolysis if we move from glucose to pyruvate, and it will become gluconeogenesis if we move from pyruvate up to glucose. But not all the steps here are reversible. So, three steps are irreversible, which are different in gluconeogenesis than glycolysis, except three steps. These steps include here the first step of gluconeogenesis, that is pyruvate to phosphoenol pyruvate conversion, and second one includes the fructose 1,6-bisphosphate to fructose 6-phosphate conversion, and the last one include the glucose 6-phosphate to glucose conversion. These three steps can't be reversible in glycolysis, so we have to take a different route than glycolysis in gluconeogenesis. So for the conversion of pyruvate to phosphoenol pyruvate, we, instead of pyruvate kinase which is used in glycolysis, we take the route which contains pyruvate carboxylase, malate dehydrogenase, and phosphoenol pyruvate carboxykinase. In the second reaction, that is the conversion of fructose 1,6-bisphosphate to fructose 6-phosphate, instead of phosphofructokinase 1 which is used in glycolysis, we use fructose 1,6-bisphosphatase. While in the third reaction, which is the conversion of glucose 6-phosphate to glucose, instead of hexokinase which is used in glycolysis, we use glucose 6-phosphatase in gluconeogenesis. Now a question arises, if we move from pyruvate to glucose in gluconeogenesis, then where do we get the pyruvate, the sources of pyruvate? We get it from lactate. Lactate is produced by the exercising skeletal muscles. And how does lactate give us pyruvate? The lactate is acted upon by lactate dehydrogenase, which forms a molecule of NADH and converts it into pyruvate. So the oxidation of lactate produces pyruvate. Secondly, amino acid. Amino acid produce pyruvate in the tissue protein hydrolysis as occurs in extreme starvation. All the amino acids except the leucine and lysine, which are the ketogenic amino acids, produce the pyruvate. Now the most important amino acid among the 18 amino acid is the alanine. The alanine combines with alpha ketoglutarate in the presence of alanine transaminase and pyridoxine, which is vitamin B6, and converts into pyruvate, while the alpha ketoglutarate is converted into glutamate. So the third source of pyruvate is the fats. Fats during starvation, as we know that glucagon increases, insulin decreases, epinephrine and adrenocorticotropic hormone increases. All these causes the increase in TAG's breakdown. TAG means triacylglycerol breakdown. Now when the triacylglycerol inside the adipocytes are acted upon by hormone sensitive lipase, whose activity increases by the effect of all these hormones, the triacylglycerol breaks down into 3 fatty acids and 1 molecule of glycerol. These fatty acids produced undergo beta oxidation to form acetyl-CoA and ATP, both of which are utilized in malate shuttle, which we are gonna discuss next. Now in the glycerol produced undergo oxidation and phosphorylation. First it undergo phosphorylation to convert into glycerol 3-phosphate. Then it undergo oxidation to convert into dihydroxyacetyl phosphate, which is a glycolysis intermediate, so can be used in gluconeogenesis. And other additional role is the oxaloacetate, which is a Krebs cycle intermediate and can be used in gluconeogenesis. Now about the 3 irreversible steps, we move from top to bottom for the steps in glycolysis. So the first step in the gluconeogenesis will be the last step of the glycolysis, which is the pyruvate to phosphoenol pyruvate conversion. Now since this is the first hindrance in the process of gluconeogenesis, so we use a different method from what is used in glycolysis. So here, instead of pyruvate kinase, which is used in glycolysis, we use malate shuttle. Now what is malate shuttle? The pyruvate, we have already talked about the sources of pyruvate. Pyruvate inside the mitochondria is acted upon by an enzyme called pyruvate carboxylase, which requires 3 factors as A, B, C, that is ATP, biotin and carbon dioxide. This pyruvate is given the carbon dioxide by pyruvate carboxylase and converted into oxaloacetate. Now as I have explained earlier, oxaloacetate can be used in gluconeogenesis, but the problem here is that it is inside mitochondria and it cannot cross the inner membrane of mitochondria. So this oxaloacetate is then acted upon by malate dehydrogenase and reduced to malate. Now this malate can easily cross the mitochondrial membrane, and then as we need oxaloacetate, so this malate is again acted by the enzyme malate dehydrogenase and oxidized to oxaloacetate. Here it is reduced and NADH is utilized. Here it is oxidized to oxaloacetate and NADH is formed. This NADH is used in the further steps of gluconeogenesis, that is the conversion of 1,3-bisphosphoglycerate to glyceraldehyde 3-phosphate. Now this oxaloacetate is acted upon by a special enzyme known as phosphoenol pyruvate carboxykinase. As the name indicates, it when acted upon by the oxaloacetate, it will remove the carbon dioxide and remove the phosphate group from the oxaloacetate. So when it does this, it forms the phosphoenol pyruvate. Now a special thing here is that phosphoenol pyruvate is also present inside the mitochondria. So it is transported via special transporter into the cytosol, which is also gathered here. Now a very important phenomena that occurs during fasting is the increase in lipolysis, that is the breakdown of fats. So this causes an increased fatty acid amount in the liver, and this increase is so much that it exceeds the beta oxidation capacity of the liver. So the extra acetyl CoA present in the liver causes a positive effect on the pyruvate carboxylase, that is it enhances the activity of pyruvate carboxylase, while it diminishes the activity of pyruvate dehydrogenase, which causes the oxidation of pyruvate as occurs in the glycolysis. So it is causing a negative feedback on the enzyme of glycolysis and positive one on the pyruvate carboxylase. So we can say that it is diverting pyruvate to the side of gluconeogenesis. Now after the first step, the next step is reversible. So phosphoenol pyruvate will converted into 2-phosphoglycerate as in same as glycolysis. Then this 2-phosphoglycerate will get converted into 3-phosphoglycerate. Then it will get converted into 1,3-bisphosphoglycerate and 2-ATP will be utilized, and then it will be converted into glyceraldehyde 3-phosphate by the utilization of 2 NADH molecule which were also formed here. So when the G3P is formed, it gets interconverted into dihydroxyacetyl phosphate. I have already told that the oxidation of G3P produces dihydroxyacetyl phosphate. Now both of these combine together. It is also a reversible reaction, so combine together to form the fructose 1,6-bisphosphate. Now another problem arises from here, that is the conversion of fructose 1,6-bisphosphate is a irreversible reaction in glycolysis. So this is the second point where we have to take a different route than glycolysis for the gluconeogenesis. So the second reaction, that is the conversion of fructose 1,6-bisphosphate to fructose 6-phosphate. We use fructose 1,6-bisphosphatase here. Okay, it occurs only in liver and kidney, and the important thing about this reaction is that it is the rate limiting step. So what happens? The fructose 1,6-bisphosphate is acted upon by 1,6-bisphosphatase. As the name indicates, it will remove a phosphate group from this and convert it into fructose 6-phosphate. Since it is a rate limiting step, it is tightly controlled. Now here is a good summary, that is glucagon, which is produced during fasting, and the gluconeogenesis also occurs in fasting, and the ATP, since a lot of glucose is and lipolysis is occurring during the starvation and fasting, so ATP, that is the energy rich state, both of these causes a positive feedback on the fructose 1,6-bisphosphatase allosteric regulation. So these causes a positive effect on the fructose 1,6-bisphosphatase, enhancing its capability, while the insulin, which is produced in well-fed state, and in well-fed state gluconeogenesis does not occur, and AMP, which includes the energy poor state, means to say that there is not in fatty acid breakdown as occurs in well-state state. And another special thing, that is fructose 2,6-bisphosphatase. It is very special factor of regulation. All three of these factors causes a negative effect on the fructose 1,6-bisphosphatase, diminishing its abilities. Now, how does these affect the fructose 1-bisphosphatase? For this, I have taken a diagram from Lippincott. Now, see here. We'll start from glucagon. When the glucagon acts on the receptor over the cell membrane of the cell, it causes the activation of adenyl cyclase, which produces second messenger known as cyclic AMP. This AMP causes the activation of protein kinase C. Now in cells, there is a bifunctional enzyme, means to say that it contains a complex contain two different types of enzyme, that is phosphofructokinase 2 and fructose bisphosphatase 2. Okay, so when the protein kinase activated and acted upon this bifunctional enzyme, it causes the activation of fructose bisphosphatase, which removes the phosphate from fructose 2,6-bisphosphate, and causes the inactivation of phosphofructokinase 2. So the we can say that the high level of glucagon is in turn activating the fructose bisphosphate and phosphatase and inactivating the phosphofructokinase. So when this one is activated, that is a phosphatase, it breaks down the fructose 2,6-bisphosphate into fructose 6-phosphate. Now, as I have explained earlier, this fructose 2,6-phosphate is very special because it causes a direct negative impact on the fructose bisphosphatase, which is used in our second exception of gluconeogenesis. So when the level of fructose 2,6-bisphosphate is decreased, it in a way causes a positive impact on the conversion of fructose 1,6-phosphate to fructose 6-phosphate. So the when we give a total overall view, the insulin causes the reverse of it, and thus activating the phosphofructokinase and inactivating the fructose bisphosphatase 2, which increases level of this fructose 2,6 bisphosphatase and thus causes a negative impact on the fructose bisphosphatase. So insulin downregulates the enzyme while the glucagon upregulates, and further study we can see that the AMP also upregulates while the ATP downregulates it. Now that explain all the allosteric effects. Now moving towards the further step. Now we have done that the fructose 1,6 bisphosphate is converted into fructose 6-phosphate. Now the next step is also reversible, that is fructose 6-phosphate conversion to glucose 6-phosphate, and the last exception of the gluconeogenesis from glycolysis is glucose 6-phosphate conversion to glucose. Now in this process we use a 2-protein complex inside the endoplasmic reticulum of liver and kidney only. This can occur only in liver and kidney. Now this 2-protein complex inside the endoplasmic reticulum is glucose 6-phosphatase, which is an enzyme which removes the phosphate from the glucose 6-phosphate, and a translocase which is used for the transfer of glucose 6-phosphate inside the endoplasmic reticulum where the enzyme is present, and the transfer of phosphate which will be removed from the 6-phosphate to convert it into glucose outside the endoplasmic reticulum. So this glucose 6-phosphate acts upon the glucose 6-phosphate and converts it into glucose while the phosphate is removed, which is then moved out of the endoplasmic reticulum by the translocase. Now a summary of the reaction include that per glucose, for the formation of a single glucose via gluconeogenesis, 6 high yield energy phosphate bonds are cleaved and 2 NADH oxidation occurs. So the last step of gluconeogenesis is also complete, and we have got glucose which is moved to the blood to increase the blood sugar level. And that was all from the gluconeogenesis. Thank you.